Most women become estrogen deficient at some point in their lives, usually due to natural menopause or undergoing surgical removal of both ovaries. Estrogen deficiency is linked to an increased risk for developing affective disorders like depression and anxiety. One possibility for this is because estrogens exert neuroprotective anti inflammatory effects by acting on microglia, the primary immune cell of the brain. Excessive and long term activation of microglia is a factor that can contribute to an increased susceptibility to mood disorders. Given this, the objective of my study is to determine how estrogens can regulate microglia and to investigate the neuroimmune mechanisms behind the behavioral and neuroinflammatory changes following estrogen deficiency. 4-month-old C57 black 6 mice were either overreactomized or received a sham operation. One half of the overreactomized mice were administered 17 beta estradiol and the other half a vehicle injection of sesame oil. These injections were given every 4 days to mimic their estrus cycle. 40 days later, the mice were injected with saline or lipopolysaccharide, LPS, to induce an immune response. We verified successful overreectomy and LPS injection in the first panel of graphs. In the 24 hours following the LPS injection, a series of behavioral tests were conducted. First, the open field and juvenile social exploration tests were performed to investigate anxiety-like behaviors. In the open field test, we saw that overreactomized mice tended to spend less time in the center of the open field, indicative of greater anxiety-like behaviors. In the juvenile social exploration test, overreactomized mice spent about half as much time investigating a juvenile mouse as compared to the control mice, and these deficits in sociability were not rescued by estradiol replacement, indicating that other hormones may mediate these social behaviors. Lastly, the sucrose preference test was done to observe for differences in depressive-like behavior across the groups. All groups had a strong preference for sucrose water at baseline, but after an immune challenge, the overreactomized mice had an exacerbated sickness response since they had no clear preference for plain water or sucrose water. We also assessed hippocampal gene expression in a subset of mice and noted that there was significant upregulation of classical pro-inflammatory genes in response to LPS. Of note, IL-6 expression was amplified in overreactomized mice and partly rescued by estradiol replacement. We also investigated expression of two anti-inflammatory microglia-specific genes, and these were significantly downregulated in response to LPS. Altogether, estrogen-deficient mice tended to have greater anxiety-like behaviors and exacerbated sickness response and amplified expression of IL-6. Ultimately, we hope that these results from this study will bring us one step closer to understanding the neuroimmune mechanisms that drive the development of affective disorders in estrogen-deficient women. Thank you.